Hello, everyone. My name is Martin, and first of all, I would like to thank the organizers to allowing us to present in this way. I hope everyone is well, and today with Primoz, we will give you a brief overview of our algorithm for efficient interactive matching of single cells and cell types. Single cell transcriptomics is a field that matured recently and has become a keystone in cancer research, personalized medicine, and investigation of cell development and differentiation. Differing from more classical bulk sequences, where we mix a large number of cells from a tissue and obtain an average transcriptomic profile, in this case, we are able to track gene expression profiles for single cells, which is critically important for disease progressions, especially in complications like cancer, where a mutation in single cell can disrupt the tissue and lead to unfavorable clinical outcomes. It is a field that has evolved substantially also in a computational sense. However, we feel there is still a paucity of explainable models that would bring the data closer to decision makers, be it data scientists or more non-data scientist type experts. It's interesting to consider the skills that a typical computational biologist or bioinformatician would use on a day-to-day basis. This would involve more nitty-gritty computational science type of skills like programming, high-performance computing, developing different pipelines, to statistical analysis like normalization and getting rid of the batch effects, to the core domain knowledge that is needed to make sense of this type of data, including cell biology, knowledge of different types of diseases and the immune system, as well as basic understanding of gene expression programs that underlie cellular differentiation and development. Last but not least, we would like the end product of analysis to be visually appealing and serve a clear message where one connects different pieces of evidence into a conclusive story and the outcome of the analysis. There has been an explosion of computational packages and libraries that are typically delivered in form of computational notebooks, providing an very interactive and reproducible type of analysis that still requires knowledge of coding of different programming languages like most typically R or Python. In our case, we would like to complement these approaches by taking a non-coding approach and build on our more than 20-year effort in delivering Orange, a highly interactive visual programming tool for data mining, machine learning, and exploratory data analysis. It is based on constructing workflows that are composed of widgets, which are concise and self-sustained components which perform computation, visualization, or training of the models. It enables fast and reproducible research that can easily be shared through communication via workflow that are highly reproducible recipes how to analyze a certain data set. Single cell Orange, or SC Orange, is a spin-off on the classical Orange data mining suite, which builds on, which delivers this classical analysis needed in the single cell transcriptomic space, and where the key data type to be analyzed are vectors of gene expressions of single cell, consisting of hundreds to thousands of quantitatively measured gene expressions. And here, Primoz will present in detail a type of the analysis and showcase our algorithm for efficient matching of single cells and cell types.
Okay, so SC Orange allows various types of analysis. So it enables clustering and classification. It allows some kind of functional analysis. It it can help us with discovering new cell types in the data and also mapping of new cells to the existing landscape. In our paper, we present a new algorithm for annotating single cell data with the cell type. And here we will present a short background on the method. So first of all, we load the data and we can observe them in the data table. Here, each row represents one cell, and so informations in the right part of the table are gene expressions. And also in case of human pancreas data, which we will see today, there is already cell type annotation present in the data, so which is the first column in the table.
And here it is important to know that this annotation was done manually by the original authors of the data by considering previously known marker genes.
So then, for example, we can map the data on the two-dimensional landscape with t-SNE method. So we got high-dimensional data on the input and we map them on 2 dimensions. And usually in t-SNE plot, groups of cells would form. So each dot is one cell. We, for example, then can select some marker genes. So genes that represent, in this case, 2 genes that represent alpha cells, for example. And in the t-SNE plot with yellow, we see cells where those 2 genes are highly expressed. So those cells are likely alpha cells. And then, for example, we can also mark some different genes like insulin gene, which is commonly observed in beta cells. And we can see part of the plot where insulin is presented, insulin gene is presented. We can, for example, also select some data in a t-SNE plot and observe, in this case, the expression of insulin. So that mean expression of insulin in selected cluster is much higher than in the rest of the data. This method enables us to discover cell types in the data, but it needs a lot of manual work. So this is the reason why we developed a new automated way to annotate the whole landscape at one shot. The method itself consists of 2 general steps. In the first steps, we identify highly expressed genes. We use a random statistic for that, for example, Mann-Whitney U-test in our case. And we select those genes that has mean rank deviation higher than selected threshold. This way, since it is done like gene-wise, column-wise, and since detected levels of genes can widely vary, this way we make sure that the analysis is made independently of mean values of gene expressions. Okay, in the second step, we then determine the cell type of cell. So this step of analysis is done for each cell separately. For each cell, we check the highly expressed genes and match them with genes that intersect with the cell type genes. And if the intersection is significant, we mark the cell with the selected cell type.
And here, Primoz,
yes.
Why it is important to do it on the single cell level?
Yeah, it is important. So some other methods do it on cluster level. And this way, some of the less represented cell types are skipped. In our case, we can identify them correctly. Okay, we packed previously described method in an annotator widget. Annotator widget gets single cell gene expression data on the input and also marker genes. Then it identifies clusters of cells and mark each cluster with the cell type according to majority voting. Here also I would like to mention that we also put some effort to make a nice whole around each cluster, that it's visually more friendly.
Thanks, Primoz. And as well as this visualization being interactive, we would like to make them as closely to the production stage as possible. Again, you can see the annotation of cell types by majority voting for cell groups of vastly different sizes. For example, for beta and alpha cells that are dominant in the human pancreas, to the much smaller groups of immune cells, for example, which you can see down on the bottom. To quantitatively evaluate the performance of our method, we compared the proposed annotation by the single cell annotator widget to the manually curated cell types by the original authors. You can see, first of all, that the cells in the human pancreas assume highly specific gene expression programs. For example, as already mentioned, the beta cells are particular in their expression of insulin, which would regulate your glucose uptake. And you can see it is a highly specific marker for the beta cells. Similarly, all of the other cells would also have a collection of their own specific marker genes that define them and are picked up automatically by a combination of the known and ever-growing gene marker databases, as well as the statistical method that Primoz presented. Quantitatively, you can see on the right that the large majority of annotator-proposed cell types are exactly matching the cell types manually curated by the original authors. And this holds for both the most prominent cells, for example, the alpha and beta cells that are in the thousands, as well as the much smaller groups of immune cells like T cells and mast cells that are present in less than 10 cases in this dataset. Speaking of the immune system, we repeated the analysis in another case study, this time concerning the immune cells in human blood. Again, our method identified the candidate marker genes that delineate these different cell types, which were originally curated and annotated by known surface markers and fax sorting. We can know that the cells here would include the predominantly cells of the myeloid lineage, the innate immune system. And in general, the marker genes that we identified roughly separate the space of cells into the antigen-presenting cells that are marked by your famous HLA, DR, and DRB genes. These are antigen-presenting genes. And then the cells, the more classical cells of the innate immune systems, like myeloid-derived cells, monocytes, and macrophages, where we can note the expression of cells that have more general antimicrobial action. And again, quantitatively, we achieve a decent correspondence with manually curated cell types with our automatic annotator method. To sum up, we presented you an efficient and scalable method for automatic interpretation of single-cell data and models that is based on statistically matching single cells with cell types, and that can be used in the future as we get more and more insight and building of single-cell atlas databases. By including it in this general orange environment, we allowed various flexible analysis and a very low burden entry to the field and a smooth learning curve for anyone interested in analyzing their own single cell data. We envision that the interactive and visually based workflows will be crucial and important tool for explainable data mining and explainable models in fields like AI and machine learning. And also envision this future work that can expand single cell orange to more recent protocols that would involve spatial transcriptomics or tissue morphology assays. We would like to conclude the talk by highlighting activities that our lab is doing to democratize data science and bring the methods closer to the wider community. On top of research project, we dedicate a lot of effort into outreach that includes workshops, blogs, and other publications. We also have our own dedicated YouTube channel that guides you to the workflows in a similar way that we just did with Primoz. Our general motivation and goal is to be able to teach a biologist or a data scientist to productively work with single-cell orange in a single day or a half-day course, which is composed of hands-out and highly interactive course materials, where one is able to grasp the key concepts in couple of hours and not be too concerned by kinks of programming, but focus on concepts more on the technicalities. And we continue to deliver these courses regularly and hopefully bring in the data science and machine learning in general closer to the scientific and non-scientific communities. To conclude, our general goals as a community are the democratization of publicly developed data science methods that is done through interactive analysis that focuses on storytelling, discussion, model critique, and general analysis of the data, that puts a heavy emphasis on the reproducibility, as well as fast and streamlined training of scientists. Finally, we envision the interactive and highly visual workflows to be a key component in future efforts for explainable AI, explainable statistics in fields of text mining, machine learning, bioinformatics, and more. I would like to thank all the collaborators that contributed to this effort over the last 20 years and more. And with Primoz, we thank you for your attention and would be happy to answer any questions or hear any comments. Thank you and have a good rest of the day.